Hello, I am Santi Dominguez, CEO of Mestrelab Research. In this video, I am showing you the presentation that I gave at the ACS Philadelphia Fall National Meeting on the 22nd of August 2016. The title of the talk was Facilitating the Inclusion of Analytical Raw Data in the Submission and Review Process. And I will start, first of all, by telling you a little bit about Mestrelab, in case you don't know the company yet. I will then talk about some of the challenges in the science publishing process, at least in what relates to chemistry and to primary research data within chemistry. I will talk about some initiatives that are currently ongoing and some of the barriers to these initiatives. And then I will be telling you about what we at Mestrelab are doing about this, about some of the tools that we have built for authors, publishers, reviewers, and readers. And finally, about the current status and future developments relating to those tools. So Mestrelab, first of all. Mestrelab was a spin-off of the University of Santiago de Compostela, the Organic Chemistry Department. It was actually founded at the end of 2004, started trading in 2005. And it is located where you can see the bigger M on the map in northwest Spain, Santiago de Compostela. The other red M's show you where we have presence at the moment, and the green M's are our major distributors. So we can give you local support in many of these areas, and of course, if you are somewhere else, we can also give you support from our head office. As a company, we started focused on the development of software to process, analyze, and predict high-resolution NMR data. This software was initially called Mestrelab, and then in 2007 it became Mestrelab. And since then, we have continued developing software for other applications. For example, processing and visualizing GCMS and LCMS data, and then more advanced applications such as automatic structure elucidation, automatic structure verification, quantification tools, databasing, and prediction of physicochemical properties. We have also not long ago released our web-based electronic laboratory notebook, MBook. The company has 30 researchers who are basically dedicated to designing and developing software. Okay, so when we look at publishing in chemistry specifically, one thing that I think the whole community agrees on is that the preservation and sharing of primary research data would be desirable. And this is for many reasons. The first one is integrity. We can prevent fraud if we have access to the primary data that was run experimentally prior to a publication. And you know, there have been some notorious cases of what could potentially be perceived as fraud. For example, you can read about an interesting case with hexacyclinol if you go to Carlos's blog, to that link there. And there are also some papers from, for example, Guido Pauli or from Nicolau and Snyder, which talk about errors in the assignment of structures in literature. I think the Nicolau paper in 2005 identified about 4,000 misassigned structures which are being published. So integrity is obviously the first and most important maybe, but replicability or reproducibility is also important. When I'm reading an article, it would be great if I could reproduce the work that the author did so that I could then arrive at the same result. The reuse of the data can be very valuable, you know, so I may be doing my own research and have my own experimental data now, and I could, for example, use data in literature to compare to my data that I have just acquired. That again could have a lot of value. Interactivity, it is a lot more interesting to be able to work with real data than to look at images, etc. And finally, if we had the primary research data available, we could also use it to build a knowledge base so that everyone could benefit from the knowledge that we can gain from data mining this data. So I think that every stakeholder will benefit. The academic institutions and funding agencies will benefit, the publishers will benefit, and both the researchers, both as authors and as readers would also benefit from being able to do this. However, we are not doing it at the moment, or let's say that in some cases we are, but this is not very common. When you look at what we're publishing in chemistry today, most of the time, I think all the supplementary information that is provided with articles is probably in PDF form. In some cases, it may be some image files, and sometimes it can be text lists like the one you see here at the bottom, where I am reporting a proton NMR spectrum, but just as a list of measured multiplets. This doesn't really tell me that much when I look at it. And it has obvious limitations. First of all, it is very difficult to detect tampering. So if there were some additional peaks in that spectrum you can see there, which I had chosen not to show, in a PDF it's very difficult for that to be identified. And nobody else can reproduce my results unless they rerun, for example, my synthesis and produce the same compound. And there is very significant information loss. Just imagine, for example, that there is some valuable information about impurities in that spectrum, but they are below the threshold that I show in the image or below the threshold which is reported in the multiplet list. So there is no chance to benefit from that additional information or even to benefit from the possibility to apply better analysis algorithms to the same data further in future. This is another example. This would be an assignment table that shows me the assignments between a set of NMR spectra and a molecular structure. And again, this is very dry compared to the possibility of looking at the structure and the spectra and see them interact together. So we have probably what could be called a problem or a limitation. And there are some initiatives to improve this situation. I think in this area, the crystallography community are an example to everyone. And upon the discovery of manipulated data, they have actually recommended that safe data be accompanied by structure factors in order to reduce the possibility of fraud. This is a great initiative. Even us at Mestrelab, we've done some work, for example, with Tony Williams at the Royal Society of Chemistry and a company called NextMove Software, to try to take NMR text descriptions like the one you see at the bottom here from the literature and to generate a spectra that people can look at. Now, of course, this is limited in that we can only generate the spectra with information that was originally reported. So this is more a visualization added value than actually providing all the information, which would be much better. So if we all agree that we would all benefit from publishing primary research data together with our publications, and we are not doing it, why is this? Why is this not happening? When I talk to people in the community about this, I normally get pretty much the same answers all the time. And the most common by far is that this is very labor intensive. Authors who publish articles in peer review publications will tell you that normally there is already a lot of work involved in generating the relevant formatted process data. So if I want to publish a patent or I want to publish an article, the supplementary information and the preparation of the supplementary information takes a long time. It is a lot of effort. If I had to provide the primary research data too, that would mean even more work. A second reason can be the lack of a common format. So if we are going to provide this primary research data, which format should that be in? And for me, that one really takes care of itself. It should be the data as it comes out of the instrument it was measured on. That is already processed or massaged to a point, but it is the format that gets us the closest to our aim of getting the data as it was acquired when the experiment was run. And finally, another reason could be the accessibility to the data by reviewers, publishers, editors, readers, and authors. You know, if we don't have a common way of looking at this data, it may be that some people along the process don't have access to it, and then the whole system wouldn't work. So these are some of the problems that we have tried to address with the project that we have internally in Mestrelab called Mpublish. What we're doing here is basically to use modern software tools to try to facilitate as much as possible the generation of patent or supplementary information quality ready materials. What this means is that we try to make as easy as possible preparing that documentation that has to be provided with the article already today. In addition to this, we also try to make it as easy as possible to attach primary research data to that. So we're basically trying to take away that objection of the fact that this will be very labor intensive. In addition to this, we want to provide free access to all researchers and even to the public in general to the primary research data that could be made available in this way. So we want to provide a tool that makes sure that anyone who wants to review that primary research data has the ability to do so. And then, thinking forward, we also want to provide tools to leverage all the possible knowledge and information we can take out of the primary research data to build better tools to support scientists in the future. So let me tell you a little bit of detail about how this works. And when I think about this project, I think about the people that would use the tools. So I kind of break it up in my head in, what does this do for authors? What does this do for publishers? What does this do for reviewers and readers? So when we think about the authors, there are several tools: spectral database, documentation writer, and the data packer. And I will talk about all these as we go, so there is one slide for each. So first of all, the database. Now, just to make things clear on how this might work, this is a tool that authors would use to keep their analytical information organized. And to be able to use this, they will need to have a Mestrelab license and a Mestrelab database license. What the database is an environment that will basically take a set of analytical raw data files and chemical structures that may come from different instruments, NMRs, mass specs, etc., and allows the researcher to build databases where all that analytical data is searchable and manageable. The analytical data can be stored and can have a direct relation to a compound or a reaction or a set of compounds, for example, in a mixture. It can be searched. This search can be driven by metadata, for example, an IUPAC name, or the name I've given to the compound, the chemist name that ran the reaction, a date, etc. But it can also be searched by chemical structure, by substructure search, or even by analytical features, such as, for example, chemical shift positions, multiplet types and positions, retention times, etc. In addition to storing, managing, and searching, the data can also be shared, and what we have added to this is that it can be one-click published at the end. So let me show you how this works. I'm now in the Mestrelab environment. I'm connecting to my database that I have built in the past, and this is where I'm keeping all my research, which I'm keeping organized, as I know that later on I may need to get back to it, or I may want to publish it, for example. And what I'm doing now is I'm using the molecular editor in Mnova to draw a substructure that I'm now going to search in the database. So I'm asking the database to return me any entities or any molecular structures which contain this substructure. And as you can see, the results that I get have all the analytical data attached, NMR and mass spec, as well as a huge amount of additional information that I saved at the time. So all my information relating to this compound and all the analytical data is organized and kept together. This is very valuable for me to review my research, but also it will greatly facilitate the publishing of it. And for that purpose, we have built another tool which we call DocWriter, which is part of the Mestrelab documentation toolkit. So again, authors that have a Mestrelab license would be able to use this functionality. And what the documentation writer does is to take a hit list like the one you just saw me produce in Mnova database, or to even take, if I don't have the database, to take a set of process Mnova files that I may just have on the disk, and it will bring together all that information to generate an ODT document, which could be in patent or supplementary information format. I can generate these with minimum effort. You will see in a minute is pretty much one click. And I can do some things along the way as well. So for example, I can even include validation tools. I could check the NMR and the mass spec match my structure, for example. I am, of course, because I'm generating this automatically, I am, of course, avoiding any kind of typing or transcription errors. And I can support specific publication formats, JAX, organic letters, etc. So back into Mnova, I assume I have an Mnova license. I call the documentation toolkit. And as you can see there, I can choose whether I'm going to go for patent or supplementary information. I can enter some metadata that will be attached to the supplementary information or patent that I generate. And in this case, I'm going to generate my publication from the database. So I ran a search for the name of my project. And in this case, I get about 30 compounds, which, as you saw earlier when I showed you the database, will have all the analytical data, etc., organized. I choose the ones I want to publish, and the software will, after one click, generate, in this case, a preview of a patent which is pretty much ready to be published. Okay, so here I have all the information I would expect in a patent. I have an index. I have the name of the different compounds as examples. I have the structures, and I have the analytical information, etc. If you remember, one of the objections was that already what needs to be done today when preparing supplementary information or when preparing a patent takes a lot of time and effort. And this has basically reduced it to the software doing the work rather than me having to spend an inordinate amount of time copying and pasting information into a Word document. This Word document we have generated here has about 60 pages with 28 compounds. 56 pages, and it has taken literally a minute to generate. You can see that we have some more tools, for example, for flagging things with color labels that may not be quite right. In this case, for example, this compound failed on mass spec, so the mass spec doesn't match the structure that I have proposed. So you can even build more intelligence into this. And we basically generate an ODT file rather than a PDF because we realize that the automation can take you 95% of the way there. But before you publish, you may want to add some further information we are not able to provide automatically, or we don't even have available. So that is how the doc writer works. And an author, by having a license to our software Mestrelab and maybe a license to the database plugin of the software too, would be able to generate the supplementary information and or patents fully automatically, as you can see. So that takes care of saving people time when generating the information they are being asked or they have been asked to produce for a long time already. That is the first wing. But our objective here really is to also be able to add the primary raw data or the primary research data to that same publication. So when I upload my article to the publisher and I upload my supplementary information, or when I present my patent, it would be great to be able to also add the primary raw data to it. And we also have developed a tool to do this as easily as possible. We call this Data Packer. And what Data Packer does is it takes a doc writer generated document, so for example the patent you saw earlier or supplementary information, and it will take all the analytical primary raw data that was used to generate that document and put it into a zip file ready for submission. This is again an automatic process which generates a single zip file, so it is ideal for upload to the publisher's website, submission website. And also it generates a single data structure, which is always the same, so it is pretty standardized, which therefore makes it easier for people to know what they are supposed to be looking at later. And it allows us to make all the primary raw data very easily available to anyone interested in the publication. So let's take a look at how this works. I'm going to repeat the process that I did earlier. So again, I'm going to use the doc writer first of all. In this case, I'm going to choose the organic letters format for supporting information. And the difference is that before I generated my document from the database, now I'm assuming I don't have a database. All I have is a bunch of Mnova files saved to my disk. I could choose which types of spectra, for example, I want to generate. And again, I have found about 20 different compounds with analytical data in the disk. I click preview, and this will generate the supplementary information in organic letters format with all my NMRs and mass specs properly organized, labeled with figure names, etc. As I did before, once I check that preview, I can generate a new document, which will be an ODT document, remember, so that you can still make changes prior to going to PDF. And the one difference with the process I showed you before is that at the end of it, I also get asked here, do you want to create a zip file with the raw data that was used to create this document? So I say yes, and basically the software knows where all the raw data is. That has generated the images that have gone into the supplementary information, and it will basically collect it all, gather it together, and put it into a zip file. It will not only do this, but it will also organize it within the zip file so that the contents of the zip file will match those of the supplementary information. Where in the supplementary information a compound was given a name, in the zip file it will have the same name as you have here, and inside it all the raw data will be available, but instead of just being labeled obscure things like 10, 14, 100, which are the folder names that come out of the instrument, is also name things that are understandable, such as, for example, proton, HMBC, mass, etc. And I also get the mol file for my chemical structure. So all this data is now available and can be easily submitted to the submission website for the publisher. So this basically takes care of what the authors need to do. They have just saved a huge amount of time when preparing this, for example, the supporting information. In addition, and with very little additional effort, they can also provide the primary research data. So those problems or objections are taken away, and there is actually a lot of added value that authors are already getting by using these Mestrelab tools. Now, just to make it clear one more time, they would need to have Mestrelab licenses in order to do this, and if they want to benefit from the database, which I think adds massive value, they would need to have a database license too. So let's assume that I am an author and I have uploaded my supporting information together with my article, and I have also included the primary research data that was automatically packaged for me. So what happens next? Okay, we have also generated some tools for publishers. And what the publisher can do, and I'll start from the bottom of this slide, is, well, actually, first of all, we assume obviously the publisher has a repository, which allows the authors to upload both articles and supporting information, and also the primary research data that goes with the supporting information. It allows them to attach some metadata at the point of upload, and it allows them to make that data available to reviewers and readers. So once that is the case, then the publisher can do a couple more things. And the first thing that they can do, if you look at the tool at the bottom, is they can use the data signer. And what the data signer is a tool that allows the publisher to digitally sign the raw data that has been uploaded by the author. This can be done manually or can be done through a web service or automated process. And the importance of that is that once that data is signed, we know that it's coming from a participating publisher. And this is where reviewers and readers will benefit. Any reader, for example, that wants to look at that raw data but doesn't have a software tool that allows them to do so can now download a version of Mnova or the full version of Mnova, but doesn't need a license to be able to read signed data. So provided that the data has been signed by a participating publisher, a version of Mnova without a license will allow them to fully interact with that data. And this basically means that we now have a tool that anybody can use independently of their resources in order to review that primary research data. Basically, the input to this Mnova version would be a set of raw data which has been digitally signed by the participating publisher, like I said, and what the reader or reviewer would get is full interactivity with the primary research raw data as long as Mnova supports it. So this will be NMR, GCMS, LCMS data, and very soon UVVs, IR, Raman, etc. Now it's important to understand that this is not a limited viewer or something like that. This is a full functionality version of Mnova, but which will work for those signed data only. But the beauty about it is that if I already have an Mnova license, I can then combine that primary research data that has been published in the article with my own, since I can also read analytical data that I have generated into the same software. Let's take a look at how this works. So this is Mnova, and now I'm going to the zip file that I generated earlier with the data packer, and I can get any of this analytical data, and I can, for example, just throw it into Mnova. And in this, I'm throwing FIDs, and in this case, a .raw file with the MS. But as you can see, this is fully processed by Mnova, and I get full capabilities, so I can zoom in, out, I can change intensities. I've just corrected the baseline, and now I'm doing a multiplet analysis, so I'm extracting all the peaks and all the integrals from this spectrum so that I have the spectrum fully analyzed. I can go and look at the detail, for example, at these two doublets here. I can check the integrals, I can normalize them, etc. So it is full Mnova functionality. It will work with the 1D spectrum, but obviously it also works with 2D NMR. So again, here I'm changing the threshold, I'm doing a peak picking, and it will also work with a mass spec. Take, for example, the case where the author has not mentioned what's under those smaller peaks in the run. I could just go click on them, and I could visualize the mass spec so I can actually identify the compounds which are under those peaks. So this is a lot more valuable than getting a PDF, and it allows me to do a lot more. I can then use the full Mnova capability also for reporting and generating text lists and information, etc. So I can very easily reproduce the work that the author did for the publication, and I could actually get to exactly the same information that that author published just by using the capabilities that this version of Mnova provides. Now, just to remind you and make the point one more time, provided that this data has been signed by a participating publisher, you don't need a license of Mnova to be able to do this. So you can do this with published data fully for free. Okay, so those are the tools we have built so far. As I was also saying at the beginning, if we take this primary research data instead of images and PDFs, we also have the ability to mine this data and to use it to generate better tools that will produce better science. And I already mentioned some examples of that, what that might be, but it would be great to, for example, have databases with all the data that everyone who publishes has run in the past, and to be able to structure search it to find the related analytical data, to even substructure search it to find related analytical data to any publication that had a structure similar or containing my substructure. It would be great to be able to search NMR peaks or multiplets, 1D or 2D, to basically ask the question, I've got this unknown, for example, has anybody published any NMRs that look similar to mine? That would be very powerful. The same could be done with mass spec, retention times, and in addition to all those searching capabilities, We could also generate, for example, prediction algorithms, so we could have more data and better NMR predictions, mass spec predictions, generate virtual screening tools, prediction of physicochemical properties, etc. So basically, the better the information that we store is, the more we can do with it. So this is where we are at with this tool. And just as maybe in the way of summary, first of all, you will remember that we talk about the ability to database all analytical data and to keep it attached and associated to compounds. This is now commercially available within our software Mnova 11. And also we talked about the documentation toolkit that will generate patent or supplementary information for me with one click, and also the data packer that will wrap up the primary research data and allow me to easily attach it to that supplementary information as a zip file. At the moment, we are supporting patent, American Chemistry Society, and organic letters formats, but we are adding formats as we speak. And we also have a data signing protocol, which is a public-private key and certification tool for the publishers, which any publisher can use by just joining our scheme by contacting us. And we have at the moment initial pilots ongoing with this, with organic letters and also with Imperial College, who are publishing their own data in the repository. We have some more work going on so that we can make it even easier for people to download and review the data, but this, remember, is available with a free version of Mnova, provided that the data has been signed by the publishers. So we have made a lot of progress in this area. I think there are a lot of tools that can make the publishing and reviewing of published data a lot easier, save a lot of time to everyone involved, and provide better tools. I'd just like to finish by thanking the people who have had something to do with this work. First of all, Dominic Sweeney from UCB Pharma, who not only provided excellent guidance as what the expectation would be from people publishing from a pharmaceutical company, but also shared with us a number of tools he had already developed for this. The Mestrelab team, you can see some of them there celebrating our 10th anniversary. In particular, Agustin Barba, who's done most of the coding for this tool. And then David Martinsen and Tammy Hanna from the ACS, who have helped hugely by not only sharing, again, what the expectation of the community is and working with us on this, providing a pilot, but also making authors available to test it. And the same for Angie Hunter from Organic Letters, who we are working with on a pilot at the moment. Henry Srepa from Imperial College, who unilaterally jumped into this and has already done an implementation of our tools into his repository, so we have an excellent working example. And Jesus Varela from the University of Santiago de Compostela, who helped us a lot with the expectation on the formats that should be published. Okay, I hope you have found this interesting. If you wanted to know more about the tool, or you want to try some of the tools, etc., you can take a look at our website, www.mestrelab.com, or you could write to us on support@mestrelab.com or sales@mestrelab.com for more information. And finally, you can write to me directly on sdominguez@mestrelab.com. I hope you enjoyed the talk and that you try the tools. Have a great day.